So in this video podcast, we'll study about PCR. We'll study about the introduction to PCR, how PCR has evolved over time, what are the principles used in PCR, and what are the instrumentations used in PCR, what reagents are used in PCR, how primers are designed in PCR, and what are the types of the PCR and its application and summary. Now, first of all, we'll see what is the introduction of the PCR. PCR is basically a replication of the DNA. Just like replication happens inside the cell, we replicate the DNA inside the PCR machine with the help of the thermocycler. It is also called thermocycler. And here difference is that we use the primer and an enzyme DNA polymerase to amplify the DNA. Now, PCR full form is basically polymerase chain reaction, means amplifying a known sequence of DNA to several orders of magnitude. That is the purpose of the PCR. So basically it is a chain reaction where small DNA fragment of interest act as a template for the synthesis of new DNA strands. One DNA molecule is used to produce multiple copies exponentially in the PCR. So what is the function of the DNA polymerase here? DNA polymerase will add nucleotides to the 3' OS of this primer according to the normal base pairing rules. Now PCR typically amplifies DNA of between 0.1 to 10 kilobase pairs. So here first you will use your template DNA, then there will be first cycle, and in first cycle there will be 4 copies. In second cycle there will be 8 copies. In third cycle there will be 16 copies. In fourth cycle there will be 32 copies. In 35th cycle, and likewise 2 to the power 36, there will be 68 billion copies. So the formula will be 2 to the power n. In each cycle we will get 2 to the power n number of the DNA. So that's why it is called exponential amplifications. Now, what is the difference between in vivo versus in vitro replication of DNA? So here in cellular machinery, the replication is mainly done by the helicase. Melting of the DNA is done by helicase, while in PCR it is done by heat. The polymerizing DNA is mainly done by cellular machinery by DNA polymerase, and in PCR by Taq polymerase. Since Taq polymerase is very thermostable. Then primer provider. What is the primer provider in case of cellular machinery? Primer provider is primase. In case of PCR, the primers in the reaction mixture which we design, we just synthesizes and put inside the PCR machine. And what are the primers in the cellular machinery? It is basically RNA. And in case of the PCR, it is DNA. What is the fidelity inside the cellular machinery? That is in vivo, that is high. In case of PCR, it is less. It is prone to mutation. However, there are a lot of polymerase nowadays which are very high fidelity, and chances of errors are very less, like Taq polymerase designed by Thermo Scientific. Now, how PCR has evolved over time? PCR has evolved from 1971 by Hargobind Khorana. Describe the replication of short fragments of DNA using polymerase in vitro. In 1976, there is isolation of DNA polymerase from Thermus aquaticus, that is highly thermostable. In 1983, Kary Mullis invented PCR. PCR invented by the Kary Mullis in 1983. In 1985, Mr. Cycler was invented. And 1986, purified Taq polymerase first used in the PCR reaction. In 1988, Perkin Eimer introduced the automated thermocycler. So what is the principle of the PCR? PCR consists of a series of 20 to 40 cycles. So the average cycle of the PCR is 20 to 40. One PCR cycle comprises of 3 steps. These steps are denaturation, annealing, and extension. Temperature used and length of time applied in each cycle depends upon the TM, that is melting temperature of the primer. Now, DNA polymerase, DNTPs, and divalent cations are also responsible for the amplification of the PCR template. Now, what is the initialization step? usually required for the DNA polymerase that are to be achieved in hot star PCR. Heating around temperature like 94 to 96 degrees Celsius for 10 minutes. Then heating dissociates the inhibitor DNA polymerase complex. So that's why heating is done. Step 1 is denaturation. So first cycle event is denaturation, heating reaction mixture to 94 to 96 degrees Celsius for 30 seconds. And DNA melting by disrupting the hydrogen bonds between bases, yielding single-stranded DNA so that primers can bind there. Now second step is annealing. The reaction mixture is lowered to 50 to 60 degrees Celsius for 20 to 40 seconds. Now primer aligns to the complementary bases on the flanking region of the DNA template. Low temperature primer could bind imperfectly. High temperature primer could not bind. Okay. So annealing temperature between 3 to 5 degrees Celsius below the TM of the primer is basically used. Now step 3 is extension. The temperature depends upon the polymerase used. That polymerase has its optimum temperature at about 75 to 80 degrees Celsius. Now DNA polymerase synthesizes new DNA strands complementary to DNA template strand by adding the DNTPs. Now extension time on DNA polymerase as well as the length of the DNA fragment to amplify. The extension time depends upon that factors. At its optimum temperature, the DNA polymerase polymerizes bases thousands of bases per minute. Now, first of all, you have a template, then you will require a primer sheets, that is forward primer, reverse primer, then you will require a DNTPs, the thermal stable DNA polymerase you will require, and which these all components are added to the reaction tube. Now there is denaturation and annealing. Annealing where the primers bind. Now there is extension step where your primer just amplifies your subsequent strands. So the extension continues and it repeated over the cycles so that you get high copy of the DNA. So what are the steps in the PCR? First one is denaturation, where double stranded DNA is converted into single stranded. Second one is annealing, the primer to single stranded DNA binding. And third one is extend primer, yielding new double stranded DNA with the help of the primers. Then cycling repeats. Step 1 to 3, 20 to 40 times. So first one is 94 like 30 seconds. Second one is 30 seconds, that will be annealing. First is denaturation. And third one is extension, that is done in 70 to 30 degrees Celsius because the polymerization is mainly carried out at 72 degrees Celsius by the polymerase. So each cycle produces 2n number of copies of the DNA. So first zero cycle 1, second cycle 2, third cycle 4, first cycle 8, and next cycle 16. Likewise, it will produce 2n number of copies of DNA. Now reagent and instrumentation. So PCR basically require DNA template containing desired segments, primers, DNA polymerase, deoxynucleoside triphosphate DNTPs, buffers, bivalent cations, magnesium and manganese, monovalent cations like potassium and additives. So what is a thermocycler? Earliest thermocyclers were designed to use with the Kleinow fragments of the DNA polymerase 1. Now Peltier element modern PCR machines. The lead temperature goes about 105 degrees Celsius. And thermal blocks mainly contains 46 and 96 well. As you will open the PCR, you will see it will contain 48, 96 well formats. So how primers are designed? Primer is an oligonucleotide sequence about 18 to 26 base pair in length, provided with 3' OH for the attachment of the nucleotide bases by the polymerase. The primers need to match the beginning and end of the DNA fragments to be amplified. In PCR, both the strands will be amplified, so one primer each of both the strands must be designed. One is forward primer, beginning of the gene of interest it will bind, and next one is reverse primer, that is beginning of the complementary strand in the 5' end.